So the DNA damage response area is one area that's receiving a lot of hype at the moment. And the reason for this are the PARP inhibitors that have now entered first-line therapy for ovarian cancer. There are more to come. There are more ways to induce damage, and we want to induce damage primarily in the cancer cells and not in the surrounding tissue. We've been doing this with chemotherapy and radiotherapy for decades, almost centuries, and it is working. The new thing is that we now, rather than introducing damage that also damage normal tissue, we can stop the repair of the cancer cells so that the high levels of DNA damage already present in the cancer can be converted to lethal damage. So the idea is to identify which pathways, repair pathways, that are important for the cancer cell to survive, and then target these with new drugs. And we have done that, many others are doing that, and I believe that it is really the combination of these ones to impair many of these repair pathways that will make the real, true difference for these cancers. So this is what we are working on now, trying to see what combinations, in which cancers, and this is one of our prime areas. In my talk here at ASCO, I was talking about new targets for DNA repair, and I was talking about two of them. One is Pol theta, which is a DNA polymerase involved in alternative end joining repair, and this is also at the same time stopping homologous recombination. So this is one way of also targeting recombination defective cancers beyond PARP inhibitors, and this is a space that is fairly well occupied by industry making small molecules inhibitors. The second area I was talking about is a new target, MTHFR2, that I presented today. And this target is normally involved in metabolism. It is involved in providing 1 carbon units for uracil to be converted to thymidine to produce the nucleotides required for repair and replication. So by targeting this enzyme that is only expressed in cancer cells, we can see very efficient killing of cancer cells, but not toxicity to normal cells. These new treatments are in the preclinical stage. And what that means is that we will take them into patients, but it's very hard to predict how they will work in those patients. And we probably need to look at what's happening in the clinic and then also fine-tune our strategies in order to really make a difference for patients.